Hello and welcome, dear students. Today we are going to discuss about the topic human evolutionary genetics. The main objectives of today's lecture are to know the basics of human evolutionary genetics, to know the origins and affinities of specific human populations, to discuss about the speciation of humans and African apes, to understand the divergence times of humans from other apes, to know the genetic differences between humans and other great apes, to understand the sequence divergence between humans and apes, and finally to discuss about the genetic differences between modern human and Neanderthals, and to understand the role of the patterns of genetic diversity in human evolutionary history. Dear students, let's start with the introduction first. Evolutionary genetics is the study of how genetics leads to evolutionary change. It involves the study of the evolution of genome structure. It is the sum of population genetics and evolution. Human evolutionary genetics studies how one human genome differs from another human genome, the evolutionary past that gave rise to the human genome and its current effects. Differences between human genomes have anthropological, medical, historical, and forensic implications and applications. Genetic data can provide important insights into human evolution. Recent biotechnological and bioinformatic advances that enable us to read and analyze genomes of modern and even archaic humans already have led to fascinating insights into human evolution. Analysis of modern and ancient human genomes allow comprehensive investigations into where and when our ancestors originated and migrated, as well as how environmental factors impacted adaptation. Dear students, let's now discuss about the origin of apes. Biologists classify humans along with only a few other species as great apes species in the family Hominidae. The living Hominidae include two distinct species of chimpanzee, the bonobo pan-troglodytes, two species of gorilla, the western gorilla and gorilla, and the eastern gorilla, gorilla growri, and two species of orangutan, the born orangutan, Pongo pygmaeus, and the Sumatran orangutan, that is Pongo abeli. The great apes with the family Hylobatidae of gibbons form the superfamily Hominidae of apes. Apes in turn belong to the primate order along with the old world monkeys. The new world monkeys and others. Data from both mitochondrial DNA and nuclear DNA indicate that primates belong to the group of Urocontoglires together with Rodentia, Lagomorpha, Dermoptera, and Scandentia. This is further supported by allo-like short interspersed nuclear elements, that is SINEs, which have been found only in members of the Urocontoglires. Dear students, let's talk about the phylogenetics. A phylogenetic tree is usually derived from DNA or protein sequences from populations. Often, mitochondrial DNA or Y chromosome sequences are used to study ancient human demographics. These single locus sources of DNA do not recombine and are almost always inherited from a single parent, with only one known exception in mitochondrial DNA. Individuals from closer geographic regions generally tend to be more similar than individuals from regions farther away. Distance on a phylogenetic tree can be used approximately to indicate number one, genetic distance. The genetic difference between humans and chimpanzees is less than 2% or 3 times larger than the variation among modern humans. Number 2, temporal remoteness of the most recent common ancestor. The mitochondrial most recent common ancestor of modern humans is estimated to have lived roughly 160,000 years ago. The latest common ancestors of humans and chimpanzees roughly 5 to 6 million years ago. Dear students, let's now understand about the origins and affinities of specific human populations. Genetic data have been used extensively to study the origins and affinities of specific population groups. For example, analysis of genetic variation in Ashkenazi Jewish populations show that these populations are most similar to one another and to other populations from the Mid East and are distinct from non-Jewish European populations, although they have received some genetic contribution from these populations as well. Analysis of Y chromosome, mitochondrial DNA, and autosomal DNA support the relatively recent derivation of Native American populations from populations that lived in Northeast Asia. These data also indicate that there may have been substantial bottlenecks as the founding populations entered the Americas some 20,000 years ago. Another even more recent event is the human settlement of the Polynesian South Pacific Islands, estimated to have taken place just 3,000 years ago. Most evidence supports a Southeast Asian origin of Polynesian populations with mixture between these individuals and Melanesian populations as they spread through the South Pacific. An analysis of mitochondrial Y chromosome and autosomal variation in South Indian caste populations indicated less differentiation among castes for mitochondrial than for Y chromosome DNA. This reflects greater levels of female mitochondrial DNA gene flow among castes than male Y chromosome gene flow. Dear students, let's discuss about the speciation of humans and African apes. The separation of humans from their closest relatives, the non-human African apes, that is chimpanzees and gorillas, has been studied extensively for more than a century now. 5 major questions have been addressed. The first among that is, which apes are our closest ancestors? Number 2, when did the separations occur? Number 3, what was the effective population size of the common ancestor before the split? Number 4, are there traces of population structure, that is subpopulations, preceding the speciation or partial admixture succeeding it? Number 5, what were the specific events including fusion of chromosomes 2A and 2B prior to and subsequent to the separation? Dear students, let's talk about the general observations. Different parts of the genome show different sequence divergence between different hominoids. It has also been shown that the sequence divergence between DNA from humans and chimpanzees varies greatly. For example, the sequence divergence varies between 0% to 2.66% between non-coding, non-repetitive genomic regions of humans and chimpanzees. The percentage of nucleotides in the human genome that had 1-to-1 exact match in the chimpanzee genome was 84.38%. Additionally, gene trees generated by comparative analysis of DNA segments do not always fit the species tree. Therefore, the sequence divergence varies significantly between humans, chimpanzees, and gorillas. For most DNA sequences, humans and chimpanzees appear to be most closely related, but some point to a human gorilla or chimpanzee gorilla clade. The human genome has been sequenced as well as the chimpanzee's genome. Humans have 23 pairs of chromosomes, while chimpanzees, gorillas, and orangutans have 24. So human chromosome 2 is a fusion of two chromosomes, 2A and 2B, that remain separate in the other primates. Dear students, let's discuss about the divergence times of humans from other apes. The divergence time of humans from other apes is of great interest. One of the first molecular studies published in 1967 measured immunological distance between different primates. The study measured the strength of immunological response that an antigen from one species, that is human albumin, induces in the immune system of another species, that is human chimpanzee, gorilla, and old world monkeys. Closely related species should have similar antigen and therefore weaker immunological response to each other's antigens. The immunological response of a species to its own antigen, that is human to human, was said to be 1. The ID between humans and gorillas has been determined to be 1.09, that between humans and chimpanzees and determined as 1.14. However, the distance to 6 different old world monkeys is estimated on average 2.46, indicating that the African apes are more closely related to humans than to monkeys. The divergence time between old world monkeys and hominoids is considered to be 30 million years ago. Based on fossil data, and the immunological distance is considered to grow at a constant rate. It is estimated that divergence time of humans and the African apes to be roughly 5 million years ago. Most scientists thought that humans and great apes diverged much earlier, about 15 million years ago. The gorilla is in immunological distances terms closer to humans than to chimpanzees. However, the difference is so slight that the trichotomy could not be resolved with certainty. Later studies based on molecular genetics were able to resolve the trichotomy. Chimpanzees are phylogenetically closer to humans than to gorillas. Dear students, let's understand about the divergence times and ancestral effective population size. Current methods to determine divergence time use DNA sequence alignments and molecular clocks. Usually, the molecular clock is calibrated assuming that the orangutan split from the African apes, including humans, 12 to 16 million years ago. Some studies also include some old world monkeys and set the divergence time of them from hominoids to 25 to 30 million years ago. Both calibration points are based on very little fossil data and have been criticized. If these dates are revised, the divergence times estimated from molecular data will change as well. However, the relative divergence times are unlikely to change. Even if we cannot tell absolute divergence times exactly, we can be pretty sure that the divergence time between chimpanzees and humans is about 6-fold shorter than between chimpanzees or humans and monkeys. One study used 15 DNA sequences from different regions of the genome from human and chimpanzee and 7 DNA sequences from human, chimpanzee, and gorillas. Chimpanzees are more closely related to humans than gorillas. Using various statistical methods, the divergence time of human chimpanzee is estimated to be 4.7 million years ago, and the divergence time between gorillas and humans and chimps to be 7.2 million years ago. Additionally, the effective population size of the common ancestor of humans and chimpanzees is estimated to be 100,000 years. This is somewhat surprising since the present-day effective population size of humans is estimated to be only 10,000 years. If true, That means that the human lineage would have experienced an immense decrease of its effective population size and thus genetic diversity in its evolution. Dear students, the shorter internodal time span, the more common are incongruent gene trees. The effective population size of the internodal population determines how long genetic lineages are preserved in the population. A higher effective population size causes more incongruent gene trees. Therefore, if the internodal time span is known, the ancestral effective population size of the common ancestor of humans and chimpanzees can be calculated. Dear students, let's discuss about the genetic differences between humans and other great apes. The alienable sequences within genomes of humans and chimpanzees differ by about 35 million single nucleotide substitutions. Additionally, about 3% of the complete genomes differ by deletions, insertions, and duplications. Since mutation rate is relatively constant, roughly one half of these changes occurred in the human lineage. Only a very tiny fraction of those fixed differences gave rise to the different phenotypes of humans and chimpanzees, and finding those is a great challenge. The vast majority of the differences are neutral and do not affect the phenotype. Molecular evolution may act in different ways, though protein evolution, gene loss, differential gene regulation, and RNA evolution all are thought to have played some part in human evolution. Dear students, let's talk about the gene loss first. Many different mutations can inactivate a gene, but few will change its function in a specific way. Inactivation mutations will therefore be readily available for selection to act on. Gene loss could thus be a common mechanism of evolutionary adaptation, the less is more hypothesis. 80 genes were lost in the human lineage after separation from the last common ancestor with the chimpanzees. 36 of those were for olfactory receptors. Genes involved in chemoreception and immune response are overrepresented. Another study estimated that 86 genes had been lost. Dear students, let's now talk about the hair keratin gene KRTHAP1. A gene for type 1 hair keratin was lost in the human lineage. Keratins are a major component of hairs. Humans still have 9 functional type 1 hair keratin genes, but the loss of that particular gene may have caused the thinning of human body hair. Based on the assumption of a constant molecular clock, it is predicted that the gene loss occurred relatively recently in human evolution, less than 240,000 years ago, but both the Vindija, Neanderthal, and the high convergence Denisovan sequence contain the same premature stop codons as modern humans, and hence dating should be greater than 750,000 years ago. Dear students, let's talk about the myosin gene MYH16. It has been evaluated that the loss of the sarcomeric myosin gene MYH16 in the human lineage led to smaller masticatory muscles. It has been found that the mutation that led to the inactivation at 2 base pair deletion occurred 2.4 million years ago, predating the appearance of Homo ergaster erectus in Africa. The period that followed is marked by a strong increase in cranial capacity, promoting speculation that the loss of the gene may have removed an evolutionary constraint on brain size in the genus Homo. Another value speculated for the loss of the MYH16 gene is 5.3 million years ago, long before Homo appeared. Moreover, caspase 12, a cystinyl aspartate proteinase enzyme, The loss of this gene is speculated to have reduced the lethality of bacterial infection in humans. Dear students, let's now talk about the gene addition. Segmental duplications or SDs or LCRs have had roles in creating new primate genes and shaping human genetic variation. Dear students, let's now understand the human-specific DNA insertions. When the human genome was compared to the genomes of 5 comparison primate species, including the chimpanzees, gorillas, orangutan, gibbons, and macaco, it has been found that there are approximately 20,000 human-specific insertions believed to be regulatory in nature. While most insertions appear to be fitness neutral, a small amount have been identified in positively selected genes showing associations to neural phenotypes and some relating to dental and sensory perception related phenotypes. These findings hint at the seemingly important role of human-specific insertions in the recent evolution of humans. Dear students, let's know about the selection pressures. Human accelerated regions are areas of the genome that differ between humans and chimpanzees to a greater extent than can be explained by genetic drift. Over the time, since the two species shared a common ancestor, these regions show signs of being subject to natural selection, leading to the evolution of distinctly human traits. Two examples are HAR1F, which is believed to be related to brain development, and HAR2 that may have played a role in the development of the opposable thumb. It has also been hypothesized that much of the difference between humans and chimpanzees is attributable to the regulation of gene expression rather than differences in the gene themselves. Analysis of conserved non-coding sequences, which often contain functional and thus positively selected regulatory regions, address this possibility. Dear students, let's talk about the sequence divergence between humans and apes. When the draft sequence of the common chimpanzee pen troglodytes genome was published in 2005, 2,400 million bases were sequenced and assembled well enough to be compared to the human genome. 1.23% of this sequence differed by single base substitutions. Of this, 1.06% or less was thought to represent fixed differences between the species, with the rest being variant sites in humans or chimpanzees. Another type of difference called indels, that is insertion or deletions, accounted for many fewer differences, 15% as many, but contributed only 1.5% of unique sequence to each genome, since each insertion or deletion can involve anywhere from one base to millions of bases. A study examined segmental duplications in the two genomes whose insertion and deletion into the genome account for much of the indel sequence. It is estimated that a total of 2.7% of euchromatin sequence had been differentially duplicated in one or the other lineage. The sequence divergence has generally the following pattern: human chimpanzee, human gorilla, human orangutan, highlighting the close kinship between humans and the African apes. ALU elements diverge quickly due to their high frequency of CpG dinucleotides, which mutate roughly 10 times more often than the average nucleotides in the genome. The mutation rate is higher in the male germline, therefore the divergence in the Y chromosome, which is inherited solely from the father, is higher than in autosomes. The X chromosome is inherited twice as often through the female germline as through the male germline, and therefore shows slightly lower sequence divergence. Dear students, Let's now understand the genetic differences between modern humans and Neanderthals. An international group of scientists completed a draft sequence of the Neanderthal genome in May 2010. The study indicates some breeding between modern humans and Neanderthals. As the genomes of non-African humans have 1 to 4% more in common with Neanderthals than do the genomes of sub-Saharan Africans. Neanderthals and most modern humans share a lactose intolerant variant of the lactase gene that encodes an enzyme that is unable to break down lactose in milk after weaning. Modern humans and Neanderthals also share the FOXP2 gene variant associated with brain development and with speech in modern humans, indicating that Neanderthals may have been able to speak. Chimps have 2 amino acid differences in FOXP2 compared with humans and Neanderthal FOXP2. Dear students, let's discuss about the genetic differences among modern humans. Homo sapiens is thought to have emerged about 300,000 years ago. It dispersed throughout Africa and after 70,000 years ago throughout Eurasia and Oceania. A study identified 14 ancestral population clusters, the most remote being the San people of southern Africa. With the rapid expansion throughout different climate zones and especially with the availability of new food sources, with the domestication of cattle and the development of agriculture, human populations have been exposed to significant selective pressures since their dispersal. For example, the ancestors of East Asians are thought to have undergone process of selection for a number of alleles, including variants of the EDAR, ADH1B, ABCC1, and ALDH2 genes. As of 2017, the single nucleotide polymorphism database DBSNP, which lists SNPs, that is single nucleotide polymorphisms, and other variants, listed a total of 324 million variants found in sequenced human genomes. Nucleotide diversity, the average proportion of nucleotides that differ between two individuals is estimated at between 0.1% and 0.4% for contemporary humans, compared to 2% between humans and chimpanzees. This corresponds to genome differences at a few million sites. The 1000 Genomes Project similarly found that a typical individual genome differs from the reference human genome at 4.1 million to 5 million sites, affecting 20 million bases of sequences. Scientists discovered evidence based on genetics studies using artificial intelligence that suggests the existence of an unknown human ancestor species, not Neanderthal, Denisovan, or human hybrid like Denny hybrid homini in the genome of modern humans. Dear students, let's talk about the role of the patterns of genetic diversity in human evolutionary history. Studies of our evolutionary history have benefited enormously from analysis of mitochondrial DNA and the non-recombining portion of the human Y chromosome. Because these two regions are not shuffled by recombination, they are transmitted intact from one generation to the next generation, revealing the paternal and maternal lineage of a population. Populations share mitochondrial or Y chromosome lineage as a result of common origins of gene flow. Comparisons of Y chromosome and mitochondrial DNA variation in a number of populations have indicated greater levels of mitochondrial DNA lineage sharing among populations, suggesting that females may have experienced more mobility during much of our history than have males. Dear students, one must exercise caution in interpreting analysis of Y chromosome and mitochondrial DNA variation because each system is essentially one highly variable locus. Statistical estimates of parameters, for example, the age of the most recent common ancestor of a group of individuals, are highly inexact. More reliable estimates of such parameters can be obtained by analyzing multiple independent autosomal loci, that is, single nucleotide polymorphisms or short tandem repeat polymorphisms. In addition, genetic data must be combined with archaeological, linguistic, and historical data to achieve an accurate interpretation of human evolutionary history. Dear students, let's have now talk about the research studies. Chinese scientists reported inserting the human brain related MCPH1 gene into laboratory rhesus monkeys, resulting in the transgenic monkeys performing better and answering faster on short-term memory tests involving matching colors and shapes compared to control non-transgenic monkeys, according to the researchers. Dear students, let's now conclude the lecture. Evolutionary genetics is concerned with the mechanisms that explain the existence and maintenance of genetic variation in traits. Evolutionary genetics benefits by providing a theoretical framework and tools to integrate individual differences and recent evolution, and thus ultimately an understanding of why we are the way we are and how we became that way. Therefore, identifying and understanding the traits or phenotypes that have been targets of selection is of major relevance to understand how humans have adapted to varying environmental conditions over time and can provide new insights into genes having played a major role in human survival. Dear students, it was all about today's lecture regarding human evolutionary genetics. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.